Hey, good morning. Miss Bittinger here, and I'm just going to go over our first set of notes with you entitled What is Science and How to Think Like a Scientist, because, we're in a science class. And of course, what we're getting ready to talk about, doesn't just apply to biology, it applies to any field of science. So that's why we had the discussion of what is science, just to get you thinking about what is science, what it isn't. So hopefully things that you put or wrote down in the discussion board you will see here during this lecture. All right, so on your note sheet, you should have what is science, and some of this is already filled out for you. So as you read through these, you should see a kind of a consensus of kind of three things that what science is and what science isn't. First of all, science is based upon data, and we use investigations and explanations and evidence to explain things that occur in the natural world. So anything that's considered supernatural or a supernatural phenomenon where we can't have data or we can't have evidence to explain that, then it's not science. So that's like number one says on our presentation, that science only deals with the natural world. We're not talking about religion. We're not talking about supernatural events, things that occur within the natural world. And notice at number two that we are going to use an organized manner of collecting our evidence and our data, because we're going to make connections between what we think is going to happen or what we think is happening, and make those connections to our data, to our evidence. Notice what I think the best part about science is that we have that tested or that experimentation of science. And number four just really sums up what science is that we're investigating, we're explaining events that occur in the natural world, and that we do that in an orderly, organized manner. So next, how is science done? Of course, we begin with an observation. This is where we gather information about events in an orderly or organized way, and we can do that with the five senses. And what we gather from an observation is called data. And then we're going to talk about two types of data. There is quantitative and then there's qualitative. And those are two different types of data. So when you think of quantitative, you think of quantity, amount, number. So your quantitative data is going to be numerical data that you gather by either counting, maybe you're counting through observation, or by some type of measurement, like maybe you're taking the mass of something, maybe you're taking the volume of a liquid, things like that. Qualitative data is going to be descriptive data, and it's going to be based upon characteristics. So when I think of perhaps a scientist that uses both, I think of perhaps Jane Goodall out in a field study, or I think of Charles Darwin. I'm sure he observed and tallied or counted certain species, but he also wrote characteristics, descriptive, qualitative data about those observations and species. All right, so the rest of the notes, we're going to compare the quote-unquote old scientific method to the new science and engineering practices. So I'm sure you've heard of the scientific method since early elementary school, and it had the 5 main steps. So we want to get out of like a step procedure that makes us kind of like a recipe, like step 1 do this, step 2 do this, and that's not what science is. Although we do have that organized and orderly manner, it's never just that cut and dry. There's a lot of movement going on, a lot of flow going on, so we just don't have that procedural like 5 steps and done method. So with the old method, we had our observation or ask a question. We formed a hypothesis. We designed a controlled experiment. We recorded and analyzed results, and then we drew conclusions. So we're still going to see some of those with our new science and engineering practices, but we're going to see more of kind of like 21st century skills tied in with these steps. So these are only read-only, so you can read through those, but notice there are more than 5. We've added in some others, and you can see that communication is a big part of these new science and engineering practices. And notice we aren't saying hypothesis, we have a new term called model. So now we're just going to compare and contrast the similarities and differences and the overlap between the old scientific method and the new science and engineering practices. So the first step with our old scientific method was to make an observation or ask a question. And again, this has to be identified. This can't be something that can't be tested. Our question or observation has to be tested or testable. Not a lot of difference between the old scientific method and the new SCPs or science and engineering practices here. It says ask a question and define a problem. But again, that question must be able to answer empirically or with evidence and data. The second step in the old scientific method was form a hypothesis. Now, we're trying to get away from that term because a lot of you like to say it's an educated guess. Well, not really. A hypothesis is much more than just an educated guess. So it's a possible explanation to a question or to a problem. Now, there are two things a hypothesis must be. A hypothesis must be testable, so I can go, or scientists can go into a lab setting or wherever they're going to test their hypothesis, and it has to be testable. So it has to be able to be have a controlled experiment done to test that hypothesis. The second thing a hypothesis must be is falsifiable. So that means it can potentially be proven false through data. Notice with the new SCPs, we do not have a term hypothesis, we have a term model. So model can mean several things. Model can be a statement, like a hypothesis. It can be a figure, it can be a drawing, it can be an actual dimensional model. As you can read here, scientific models can make the invisible world tangible and can be edited as new understandings develop. So as new technologies come about, perhaps, then we can go back and revise the model and then test it again. So again, we're going to see the overlap and the connections and maybe some similarities and differences between the old scientific method and the new science and engineering practices. So something that stays the same is designing a controlled experiment. So you may not know exactly what a controlled experiment is, so we're going to go over that. So with a controlled experiment, we want to make sure in our experiment what is giving us our data. And that is where the term variable and control comes into play. So you can read there about a variable. A variable is simply something that is changed within the experiment. And some great examples there: changing the temperature, changing the amount of light, changing the time, the concentration of the solutions, perhaps changing the acidity, perhaps changing the location, perhaps changing the pH, which would be acidity, things like that would be all variables, things that the scientist is purposefully changing. And this is where the term controlled experiment comes into play. We want to make sure that we're just changing one single variable at a time. We don't be changing multiple things because then we don't know which variable, which change gave us the results or the data that we are collecting. So within our controlled experiment, we're going to have two groups. One is called the control group and one is called the experimental group. The control group is the group that does not receive the variable that the scientist is deliberately or purposefully changing. The experimental group is the opposite of that they are receiving the variable or the change that the scientist is purposefully making. Everything else between those two groups stays exactly the same except for that one variable. So you have an example here on your note sheet that applies right now for our coronavirus pandemic. So you can read through that example, and we want to figure out 4 things about this example. We want to figure out our two groups, our control group and our experimental group, our constants, what variables were kept the same between both groups, and then what was the variable being changed. So within this example, the control group would be the group of volunteers, probably 25 volunteers, that did not receive the vaccine within their pill. So everything else was the same between that control group and the experimental group, but the control group did not have the vaccine in their pill. So the experimental group would be the group of volunteers, the other 25, that did receive their vaccine within their pill. Some things that were kept the same. So if you read this last sentence here, both groups are given a pill to take that is identical in size, shape, color, and texture. Those are constant. So between the control group and the experimental group, those things were kept exactly the same. So I'm sure you could think of other constants that would need to be the same between both groups as well, such as the age of the volunteers, the gender of the volunteers, what time of the day they took the pill, did they take it with food, did they take it with a certain beverage, etc., etc. So in this example, the variable being changed is the vaccine. All right, so within an experiment, we have two variables, and lots of times students come to me and they have been mistaught or misunderstood. They just misunderstand about these two variables. So I want to make sure we go over these, the independent and dependent variable. So the independent variable is the variable that the scientist purposefully or deliberately changes on, like they do it on purpose. So back in that previous example, the independent variable would be the vaccine. The scientist was deliberately giving one group no vaccine and then the other group the vaccine. So the dependent variable is the outcome of that change. So it's the result of the independent variable. And really it's the data the scientist is collecting. So I already went over C. It was the vaccine and the pill. So D, the dependent variable in our previous example, would be like the outcome or the health of those volunteers. So that could be measured as well. Maybe they're taking their heart rate, maybe they're taking their blood pressure, maybe they're doing observational qualitative data as well. All right, looking at scientific method for after the experiment of recording and analyzing results and comparing that with the new SCPs. Not much different here, not much difference between the two, except for here, we want to make sure that if there are any sources of error that we result those, we write those down, because we're human. Errors are going to happen, or we're going to get errors with machinery, we're going to get errors in data somehow, maybe something gets contaminated, etc. We need to write those errors down. We want to make sure we have meaning with our evidence, and what that means is we just don't get our data and say we're done. We want to make those connections back from our data to our model. We won't do math and computational thinking a lot in this class, but you'll see it again in your other classes. All right, so with the old scientific method model, drawing conclusions was the last step. This is still important. We want to see if our data supports or does not support our model or our hypothesis. So you can see we got the last steps here for our SCPs, and they again are similar to drawing conclusions, but we want to make sure our data and our evidence support our conclusion. We never want to change our data to meet our model, nor do we want to change our model to meet our data. It is okay if your data does not support your model or your hypothesis. Do not change to make it prove it. That is biased or false science. One thing we're going to try to do in person and or virtually is argumentation and communication with scientific findings. You know, it's great that you have your results and you make those connections, but we need to strengthen our evidence, or we need to strengthen our solution, our conclusion. So one way we do that is through communicating with our peers, and they can find our weaknesses and help us strengthen our reasoning. All right, so lastly, there's a video here for you to watch. I think lots of times we as humans in general misuse hypothesis and theory. They're actually two different scientific terms. So there's a little video for you to watch here, and after you watch the video, just see if you can, in your own words, explain the difference between a hypothesis and a theory. And I'll see